So we will study plant growth and development, where development is the product of growth and differentiation. It is the sum of growth and differentiation. So what is growth and what is differentiation we will discuss. So you all know that all the plant or animal develops from a zygote. Zygote is nothing but a fertilized egg. And the phase of growth is very precise. Growth is precise and highly ordered succession of events. So if growth is ordered, precise, then it generates different kind of tissues and body organs which function different from each other. Like a tissue of eye can see, like a tissue of xylem cells or xylem phloem that helps in conduction, while some tissues like pericycles, root hairs, that help in absorption. So different tissues function differently, or they are made for different works. So your cell get differentiated into tissues. That process of developing of cells in different tissues, or you can say this is a division of labor between the cells. A group of cells is defined to do a particular job or a particular work. So there are different tissues. And these tissues make body organs. This process is called differentiation. So both growth and differentiation makes the development. So in this lecture we will discuss factors which govern and control the development process. These factors are both intrinsic. Intrinsic are born from inside, and extrinsic that is born from outside. Intrinsic like hormones, plant hormones. External like light that comes from sun. So what is growth? Growth is the irreversible permanent increase in size of an organ or its parts or even of an individual cell. So growth is irreversible. Irreversible means can't be reversed. We can't come back. Permanent means not temporary. And increase in size or organ or its part or even of an individual cell. But increase in this definition is somewhere not correct, because sometimes there is no increase but still there is a growth. Okay, and we will discuss something more about plant growth and development in our next lecture.
So growth, what is growth? We previously defined growth. So growth is the collection or a group of something like increase in size, precise and correct. So what is mean by precise? First we will discuss precise and highly ordered. Precise. What mean by precise? And why we need precisement? Why it is highly ordered? We will also discuss dedifferentiation, redifferentiation and differentiation. There are three terms: differentiation, redifferentiation, and dedifferentiation. But before comparing these three, we will discuss totipotency. Totipotency. What are totipotent cells or stem cells? What are stem cells or totipotent cells? So why we need precisement? We can discuss both in plants and animals. In animals, we have these 5 fingers. Size of this thumb is not equal to this finger and not equal, this length of this finger is not equal to this one and not equal to this one and not equal to this one. If there is not a growth is not in ordered fashion, not precise, then what will happen? This hand has 4 fingers and they are all of equal length. All of equal length. But this is true? Not true. We have fingers, they have different lengths. And if we want to make a fist, we want to grab something, then these 4 fingers help in grabbing, like this. And if they have equal length, will they behave in this way? We can make a grab like this? Not. It is not possible. So, after a specified time, after a time limit, your cells stops growing. Or they go in a phase that is of resting phase. That resting phase may be lifetime, maybe sometime you can change that resting phase, you can stimulate that resting phase, and after that, that cells can grow again. Can grow again. So, why this is highly ordered? Because you can see the shape of a leaf. So these cells, we know there was a 1 cell and there was a 2-cell structure, that is zygote. 1 cell is from female parent, 1 is from your male counterpart. Then we have a zygote, and this zygote can further divide in many cells. So all of the organisms in nature are derived from this zygote. There is 1 cell, then there is 2-cell stage, then there is 4-cell stage, so on. But if you see, then this margin, if there is a regular, not in ordered growth, not precisely this cell stop to divide. If this will not stop dividing, then this leaf like this. There will be a leaf like this. If again this will not stop dividing, not stop dividing. What about these ribs? These also in ordered fashion. If you want to see a vertical or lateral section of leaf or leaf vein, then you will find like this. There is a canal. There is a canal that can carry water. Or there is some cells, green cells, which can make food by photosynthesis. But these canal cells lack the chlorophyll. They don't able to form food. So, in a leaf, not an entire plant. In an entire plant, if you want to see, then there are petals, sepals, conductive tissue. This is your flower. There is a stigma in the flower. There are anthers in the flower. Anthers in the flower. And all are composed of cells. These cells are usually colored. These are the cells of petiole, and they are usually colored because they have a colored pigment in them. They have red carotene, the colored pigment in their cells. But leaves are usually green, not of red color, because they have chlorophyll. They lack chlorophyll. They are not able to photosynthesize. So, there are different cells present in a plant at a single time, same time. So, you can see different cells can perform different duties. This part of cells that allow them to perform different duties. This cell of petiole can't synthesize food because it lacks chlorophyll, but the cell of this leaf can make food by photosynthesis. They have different functions, and they are developed, they are grown in a way that they can perform different functions. This process is called differentiation. This process is actually called differentiation. Now you have multiple types of cells which can perform their duties at same time. One cell can synthesize food, one cell can conduct water, one cell can diffuse waste at the same time. So there are different cells, and this process is called differentiation. So there are totipotent cells. What are totipotent cells? So if there are cells, this is a cell. Assume this is a plant cell, and this is performed, this is grown to make colored pigment. This is a cell of petiole. Cell of petiole. And this will generate the pigment so that it can look yellow and can attract insects. This is the duty of this cell. So that flower can look beautiful and it can attract more insects that can help in pollination. This is the primary job of this cell. And now consider, some part of the cell dies, or some part of the plant dies due to unfavorable conditions like drought, and this cell remains alive. This is the only cell that remains alive. Now this cell can divide or can make whole plant? Not possible. This can't divide. Are the cells. These are non-totipotent cells because they can't divide. If they are able to divide, if these petiole cells can be able to divide in broth or in external medium, then they will produce same kind of cell. They will produce same kind of cell. Same petiolar cells of red color or yellow color. We can't make a cell that is of green pigment or that contains chlorophyll or that of green color that can photosynthesize or then that can make food for plant. So your plant will not survive. If these cells, assume these cells can be grown into chlorophyll cells, into petiolar cells in which there is red pigment, and if these can be grown into green pigmented cells or chlorophyll cells, and other cells like xylem cells, phloem cells, which can perform different functions. Such cells are called totipotent cells. So your totipotent cells are the cells which can be grown to perform any function desired by plant. Actually, in your animals, such cells are called stem cells. And nowadays, this is very famous therapy, stem cell therapy, in which cells from placenta are taken and preserved and can be used to grow desired cells anytime when we need, or we need to implant like bone marrow transplant and other uses. We can use these stem cells. These are cryopreserved. Cryopreserved means at liquid nitrogen temperature we preserve the placenta, and that have stem cells. Actually, stem cells are totipotent cells. So in plants, we usually term them totipotent cells, and in animals we used to call them stem cells. So stem cells and totipotent cells we discussed. So if you want to see the cell cycle, then there is a resting phase, synthesis phase and dividing phase. In this phase, synthesis phase, your cell synthesizes the material required for division. So it usually doubles all its organs, all the chromatin, all the material found in cell, it gets doubled, and then it enters the dividing phase. This is of our use. Today we will discuss this resting phase. Resting phase. Some of the cells get trapped in resting phase. And they always remain in the resting phase. So totipotent cells or stem cells actually stay in resting phase without destroying their ability to differentiate. Nowadays you can see that there is a Nobel Prize in stem cell research in which all kind of cells, even skin cells, can be stimulated to form any kind of cell. So it is shown that they are all the cells are totipotent cells. But previously we observed, or we only get the stem cells or the cells from placenta are only the totipotent cells. But today we know that any kind of cell from body, even human body, can be grown to be totipotent. So this is the resting phase. If the cells get trapped in resting phase, it will not differentiate or it will not divide. Sometimes after differentiation, these cells get trapped in resting phase. Sometimes before differentiation, they trapped in resting phase. Such cells are your stem cells. Cells of placenta are cells in resting phase. They are not dividing, but they have ability to differentiate. If we will provide them some medium from outside, they will start dividing and can be differentiated from to any kind of cell. So this is your differentiation. What is redifferentiation? What is redifferentiation? Redifferentiation is the same thing when we trap cells in resting phase. They are able to differentiate and we trap them in resting phase. If we break this resting phase, we will provide some outside growth or medium so they can differentiate and can divide. This is redifferentiation. Then dedifferentiation. Is the cells already differentiated? Already differentiated. Assume these are your petiolar cells. These are of red color, and they differentiated into petioles, petiolar cells. Now they have to perform their duty. They have to attract insects, help in pollination, okay, due to bright color or bright red color. So these cells also trapped in the resting phase. They are trapped after differentiation. They are trapped in resting phase after differentiation. Now what we have to do? If you want to get some totipotent cells from these petiolus cells, then what we have to do? We have to de-differentiate those cells. We have to de-differentiate those cells into differentiable cells. So what we will do? We will de-differentiate those cells, and then we can use them as totipotent cells. So this is your de-differentiation. So now you have a clear-cut idea between differentiation, re-differentiation and de-differentiation. Okay?